Well, so I want to welcome you to the first ever Virtual Scholar Awards program. You're making history with us right now, and it feels really good. And on behalf of the Northern California chapter, thank you, San Francisco State, for being the very first of our university partners to recognize how to reimagine how to meet and greet and celebrate ARCS scholars. We didn't have a Scholar Awards celebration in the fall. You know why. It doesn't surprise me that San Francisco State is the first of our university partners to do this, because you have an outstanding leader, Dean Carmen Domingo, so deeply committed to STEM education. Her enthusiasm and her smile is both inspiring and contagious, and her dedication to young scientists makes me feel so proud to know her. Thank you, Dean Domingo. And thank you, each and every one of the ARCS scholars who's here tonight. While Zooming is not so grand as a gala celebration at a grand hotel, this meeting certainly allows us to have a moment to see one another and hear from each of you. We look forward to understanding what keeps you up at night, what your science is headed towards, and how you're going to make the world a better place. And we are so grateful to be on your team. I also want to thank a shout out to Lanny for her detailed planning, a beautiful flyer, and managing all the technical support. Lanny, we could not do this. You know, we talk about herding cats, but herding scientists is even harder. And lastly, I want to say thanks to the University Relations Council, especially to Betty Alberts and Bailey Meyer and Jan Goodson. They are your official liaisons for San Francisco State, and they are a great team. And we just look forward to making more history together. So with that, over to Chris Brent, co-VP of University Relations for our chapter. Chris.
Thank you, Joan. With all of that, there's not so much more to say. My will close the meeting, Leslie Van Houten. So we both just have the great honor to be with you all tonight and being part of this, as Joe said, first time when we've had a Zoom celebration with scholars, and also where we've done it, at least in many years, on a university by university basis. And we love the opportunity to have you all get to know each other, as well as us to get to know you, but most importantly, for us to have the opportunity to celebrate and honor you, and to have you know that we are rooting for you in every way that we can. And in addition to your award, which we hope, made you feel good and inspired you in and of itself, we want you to know that there's this group of women who are passionate about supporting you in the work that you do, the discoveries that you make, and the difference that you will have on the world. And so this is the opportunity for us to really just let you know how proud we are of you and how supportive we are of all that you're going through. And this is the toughest year ever, right? You're working solitary, you're not getting to see your colleagues as much in the labs. And I'm sure if there's ever been a year when you go, boy, being a scientist is kind of maybe a solitary thing, except that I know that San Francisco State's doing everything they can to have you be part of the community. So know that we're there too as part of your community, and we wish you the best. And you all, I saw many anyway, had their medallions on. That's just that tangible thing, especially in this year of virtualness, to say, you all are selected personally for your achievements and your potential by the university. And that means a lot. I think that you were selected by Dean Domingo and her team speaks volumes. We are so proud of the university and our partnership with San Francisco State and all the great work that students do there and go on to do beyond their time at San Francisco State. And we know that you join that group of excellence. I just want to say very briefly, U11 as San Francisco State ARCS cohorts in the class of 2021 join a full group of 79 this year across Northern California. And all together, you're part of a collegial collection of excellent students and scholars and mentors and scientists out in the real world of over 2,800 from our chapter and almost 11,000 since we were founded in 1958 in the LA chapter. So there's just a passion that we feel for what you do. And we know that you'll be out there in the real world making all the difference along with those who have gone before you. So with that, I want to turn it to Dean Domingo. We couldn't be just more delighted to partner with her in this venture of supporting scientists. She, as you all know, is a visionary in her leadership, in her commitment to science, and her commitment to equity in science, and the support that she gives the over 7,000 students that she leads and all of the faculty and administrators. So people have called you really a visionary in leadership, and we think of you as a visionary in our partnership. You do, as Joe mentioned, set the bar very high for all of our universities in terms of how closely you work with us and the kind of relationship we have. So with that, with your science career and now administrative career in 3+ years as the dean, both interim and full dean, we just want to thank you for what you do and turn it over to you to introduce your scholars tonight.
Oh my, I'm speechless. Thank you so much for your kind words, and they're incredibly generous, truly. And thank you for your leadership of the Northern California chapter of the ARCS Foundation. We're truly delighted and honored to host this year's awards celebration, and that we're the first ones to do this Zoom event. The Northern California chapter of the ARCS has supported our master's students for nearly 20 years. During that time, you've supported 187 master's students in departments across our college. The total amount of funds raised to support these students is $1.48 million. It's truly amazing. As a non-PhD granting institution, we don't have the same financial privileges as our UC counterparts. Hence, your ARCS scholarship provides a major source of support for our master's students. And for that, we are very grateful for the hard work and dedication you and all of your members have had in supporting the next generation of scientists. Tonight, you will learn about our ARCS honorees in a lightning round of presentations, as each scholar has approximately 3 minutes to present their research project to you. We will have their research mentors, which are really the backbone to the success of our ARCS scholars. They have devoted so much time and attention training these students, so we're delighted that they are here to introduce them. They'll also ask them a follow-up question. You will be impressed with the research activities that they've been able to pursue despite the many challenges that they've had to face with the pandemic. Our academic and research programs have largely been working remotely, and hence, through creativity, perseverance, and resolve, our honorees are making progress on very exciting research projects. So with no further ado, I am pleased to invite Kevin Simonin, Associate Professor in Biology, to introduce our first ARCS speaker.
Thank you, Carmen. It is really my pleasure to introduce you to Anjum Gujral. It's been really fun to work with Anjum these past several months, and I've learned a lot working with her. Research in our lab largely focuses on species variation and plant water use strategies and how this variation is related to differences in climate. Anjum is helping our lab to think about plant water use strategies from an evolutionary perspective, which allows us to begin to consider how recent climate change may be altering long-term trends in plant physiological adaptations. But I'll let Anjum tell you more about that.
Thanks, Kevin, for that introduction, and to everyone that could be here today to not only learn more about the scholars and their research, but to also recognize the donors for their generous contributions. So today I'll be talking about my research that in general focuses on investigating coordination in plant structural and physiological traits. So to begin, I would like to give some background on what plant functional traits are and why they're important for us to study. So plant functional traits are morphological and physiological traits that influence the ability of plants to grow, survive, and reproduce. And with that being said, we can use plant traits as indicators of plant strategies for growth, survival, and reproduction. And so if you can see here on this diagram, these are some broad categories of plant traits that we're interested in studying as plant ecologists. And these arrows indicate how these broad categories are interconnected. And coordination in morphological and physiological traits is really important for us to study because these traits can influence the ability of species to grow where they do, and specifically how they might respond to climate change. And so the objectives of my research are first to determine whether physiological traits are coordinated with morphological traits. And I'm going to be doing this across a diverse range of species. And so you can see here in this figure to the right, these are a variety of different leaf sizes and their associated vein densities. And secondly, I would like to investigate whether trait coordination is phylogenetically conserved. And so here I'm interested in asking whether this coordination has remained relatively constant throughout evolutionary time in the select lineages that I will be studying. And lastly, I would like to map the distribution ranges of these species and along with their climate variables to determine how trait coordination influences current and future species ranges under climate change. So my research is significant because plants are really the foundation of all terrestrial ecosystems. And in the face of climate change and habitat destruction, there's an increasing need for understanding how to restore and protect biodiversity. And in order to do this, we must really understand how plants function, and functional traits can help us understand that as they can inform mechanisms of plant performance. Firstly, I would like to thank my advisor, Kevin Simonin, for his unwavering commitment and investment in guiding me through my master's thesis, and especially during such a difficult time. And the ARCS Foundation for their generous contributions, as my research would not be possible without your support. And specifically the Karen and David Chamberlain Family Fund, as well as the Gil and Robert Smilik Family Fund. Thank you.
Thank you, Anjum. I think one of my favorite things about your work is that it seems really applicable to our current and future conservation efforts for plants. And I was just wondering if you had any thoughts on how this will help inform potentially future conservation and restoration efforts.
Yeah, so I am really interested in looking at conservation from an evolutionary perspective. And this is quite uncommon, but I think very important because when we look at how species will respond to climate change or any environmental disturbance, that really depends on the species' evolutionary history. So I'm really excited about being able to apply an evolutionary lens to conservation.
That's great. I'm really excited about your work. Well, I'd now like to pass the mic over to George Gassner from chemistry, who's going to introduce our next ARCS scholar.
Thank you, Kevin. It's my honor to introduce A Twin, who is currently completing her MS degree at San Francisco State University in chemistry with an emphasis in biochemistry. Through the course of her undergraduate education, A is actively engaged in biomedical research. Her MS thesis work at SFSU centers on the mechanism of protein aggregation in neurodegenerative disease. After completing her MS degree, A plans to continue to develop her skills as a scientific investigator and advance her education to an MD-PhD degree.
Thank you, Dr. Gassner, for your introduction. Hello, everyone. Again, my name is A. I am a second-year graduate student at the Department of Chemistry and Biochemistry. I am currently doing my research at UCSF, and my thesis research focuses on understanding tau protein aggregation in Alzheimer's disease. So in this presentation, I'll briefly explain about what my project is. So in a healthy brain that is shown in the left here, there are tons of billions of neurons that are responsible for critical function of our brain. However, in patients with Alzheimer's disease, those neurons are progressively damaged and killed by what are called plaques and tangles. So these are hallmarks of Alzheimer's. And my project focuses mainly on the tangles that consist of tau protein aggregates. So you might be wondering what is tau and why are they found in the tangles in the brain? Next slide, please. So tau is like any other normal proteins. It has its specific functions, specifically in the neurons in the brain. So tau is known to bind and stabilize microtubules. So you can think, and so in Alzheimer's disease, however, tau proteins break away from microtubules and form tangles. So researchers have found that those tau proteins in the tangles have excessive chemical groups on them, specifically phosphate groups, so which we call it hyperphosphorylation of tau. And so we think that those excess phosphate groups are the one what's triggering tau to aggregate and form tangles in Alzheimer's disease. So this is basically what I'm currently researching on as part of my thesis. And next slide, please. So our brain is a very complex system. It's been over 100 years since Alzheimer's disease was discovered. Since then, extensive research has been done to uncover the mystery of this disease. Yet we are slowly progressing toward finding cure for it. It's definitely a long process, and but I have to say, I'm really thrilled to be a part of this. Hopefully, our new understanding of tau tangles will aid in creating inhibitors to prevent tau aggregation in Alzheimer's disease. With that, I would like to thank all my advisors for their guidance and support on my project, and of course, ARCS Foundation and the donors for their generous support. Because of your support, I am able to do this research, and I'll be able to complete my master's degree in time by next year and further my education by applying to medical programs. So thank you so much.
Thank you, what are the prospects for the development of aggregation inhibitors as effective drugs for the management of Alzheimer's disease?
Thank you for your question, Dr. Gassner. So actually there are some inhibitors already developed to target tau protein aggregation, but they have not shown any significant improvement in the treatment of Alzheimer's disease. So I think it is because we still do not have a full comprehensive understanding of this problem and the exact cause of this disease. So hopefully my research will shed some light into the cause of this disease.
Thank you. Well, I would like to now introduce Dr. Federico Ardea from the mathematics department.
Thank you so much for the introduction. So it's my great pleasure to introduce our next ARCS scholar, Cesar Mesa. You know, professors don't exaggerate when we say that our students are our best teachers, and I have really learned so much about mathematics and about education from Cesar. Cesar Mesa grew up in Southern California, and he got his undergraduate degree in mathematics at Pomona College, and he's now a master's student in our math department at San Francisco State. His research is at the intersection of combinatorics and high dimensional geometry. And aside from that, Cesar is also really interested and very experienced in math education. And he has helped design several different initiatives and interventions that seek to build a really inclusive and equitable mathematical community. So I'm looking forward to hearing Cesar telling us a little bit about his research.
Thank you for the introduction. So in my research, I study objects called polyhedra. So in the next slide, I'll show you an example. So first, we define a polygon, which is a two-dimensional shape with straight sides and no indents. So for example, the green square on the left is a polygon. However, the object to the right is not because of the indent at the top. And so a polyhedron is a three-dimensional version of a polygon. So if you can imagine taking the square and pulling it into the third dimension, we get the green cube. However, the object to the right is not a polyhedron because of the indent at the top. So in the next slide, I'll show you some of the components of polyhedra. So here we have a tetrahedron. And so the first component of a polyhedron are the vertices, which we can think of as the corners of the polyhedron. And in this case, we have 4. Next, we have the edges, which are the lines that connect the vertices together. And so if you look closely, there are 6. There's 3 going towards the top, and then 3 at the base. And finally, we have faces, which are the flat sides of the polyhedron. And in this case, there are 4, 1 in the back, 2 in the front, and then 1 at the base. And in the next slide, we'll see more examples of polyhedra and the number of their vertices, their edges, and their faces. Some of you might know these as the Platonic solids. And remarkably, for any polyhedron in the world that exists, if you take the number of its vertices, subtract the number of its edges, and add the number of its faces, it'll always equal the number 2. And so this is a fundamental truth of polyhedrons that is at the center of my research. And in fact, if you can somehow imagine and visualize polyhedra in higher dimensions, so let's say dimension 4, 5, or even 10, similar relationships hold. And so in the next slide, I have an example that kind of shows you the application of polyhedra in the real world. So if you can imagine yourself running a small food bank in your local community, and imagine that there are 3 distinct locations, and each location has its own limit on the amount of food it can safely store inside the facility. And each facility has its own costs for staying open. And so each of those restrictions on the facilities actually describes a face of a polyhedron, which is here on the right. So if your goal is to maximize the amount of food you can distribute to your community while minimizing the cost of keeping your food bank open, you only need to look at the vertices of the polyhedron to find your optimal solution of the amount of food you can serve while minimizing your costs. And finally, I would like to thank my advisor, Dr. Federico Ardila, and the ARCS Foundation for their support. With your support, you make attending graduate school a reality for me. And finally, I look forward to attending a PhD program next year to continue my path to become a professor. Thank you.
Thank you so much for a great presentation. But I have to ask you, what about this 4 dimensions, 5 dimensions, 10 dimensions? I mean, my vision is only 3-dimensional. And geometry is supposed to be the study of shapes. So can you tell us a little bit about how do you go about trying to visualize these high dimensional shapes that you study?
Yeah, that's a really good question. It's also a very common one. So if we go back to the example with the food banks, in that case, we had a 3-dimensional polyhedron because we had 3 distinct food banks. But if you can imagine, running 4 or 5 or even 10 food banks, the way that you can visualize those objects in higher dimensions is just by focusing on one food bank at a time. So for example, if my first food bank can only store, let's say 500 meals every day. So there's a limit. So you can think of each of those food banks as having a certain amount of allowable values in the food bank. So for example, you can either store like 0 amount of food, or maybe 10 meals, or up to 500. And then in your second food bank, you might be able to store 700 meals, and so on and so forth. So you can think of each of those components as being an independent scale or slider that determines the shape of your higher dimensional polyhedron.
Very cool. Thank you so much, Cesar, and thank you also for kind of making the point that sometimes science that looks like it's really pure it actually ends up being really applicable in everyday situations.
Thank you.
So next, it's my honor to introduce Professor Erika Sanchez from our biology department, who will introduce our next ARC scholar.
Great, thank you, Federico. Welcome, everyone. Thank you for joining us this evening. It is my pleasure to introduce one of my first graduate students, Jan Kahulau. And actually, Jan is very brave. He joined a brand new lab in our department. I started my assistant professor position in January, and we went into the pandemic situation about 6 weeks in. But I did have the honor of meeting Jan in the classroom originally in my virology class in my first semester and his last. When he completed the spring semester, he graduated from San Francisco State and decided that he wanted to continue studying viruses, which is what our lab studies. We're really interested in oncogenic viruses, or viruses that cause cancer. And Jan's going to tell you about the beginnings of his master's thesis, where he's been working on a herpes virus that causes cancer. We're really interested in how this herpes virus causes different signaling within human cells and how this alters the host during infection. I also wanted to note that I myself was honored with an ARCS fellowship as a graduate student at the University of Washington back in 2010. And so it's really, really great to see one of my first students being honored with the same fellowship, as this was extremely important to my success early on in graduate school. So I thank you so much for continuing all the work that you've done with students. And with that, Jan, please share your work with us.
Hi, everybody, and thank you so much, Dr. Sanchez, for the wonderful introduction. And I want to welcome all of you to our event tonight. Thank you for coming. So once again, my name is Jan, and I wanted to start my talk off by telling you a little bit about me. So I am a first-generation Filipino-American immigrant born in the Philippines. And then when I was about 3 years old, me and my family immigrated to the San Francisco Bay Area. And I've stayed there since we moved here, where I eventually went to San Francisco State University, as Dr. Sanchez mentioned, to do my undergraduate degree. Now, unfortunately, right before I started college, my beloved mother, who I miss very much, passed away of acute myeloid leukemia. Now, I had already had an interest in biology before starting college. However, my mom's fight against cancer focused my attention on biomedical science, and more specifically, cancer biology, which is the main motivation that I've had throughout my career here at SF State, and inspired me to join a research lab. So for my undergraduate degree, I joined the lab of Dr. Lily Chen and studied the bacteria called Group B Streptococcus, pictured here on the left. And then now in my master's, we're studying a cancer-causing virus called Kaposi sarcoma herpes virus, pictured on the right. After my master's, I am planning to apply for and join a biomedical PhD program to even further my training in cancer biology and hopefully lead my own research group in the future as a PI. Next slide, please. So once again, Kaposi sarcoma is a cancer. Specifically, it is a cancer of the endothelial cells. These cells are what line our blood vessels, as you can see pictured on the left. Now, when these cells become cancerous, they divide uncontrollably and develop a tumor. Now, if you're wondering what a tumor looks like in your blood vessels, these manifest themselves as lumps or lesions on your skin, shown here on the right. Now, researchers discovered that Kaposi sarcoma is caused by a virus, as pictured on the bottom, called Kaposi sarcoma herpes virus, which I will refer to as KSHV. Now, it's not entirely known how the virus causes cancer, but the virus does code a receptor. Advance once, please. So this receptor is required for the virus to replicate and propagate itself within the host cells. This is called a G protein coupled receptor, and is a class of proteins that many species have, including humans. It is actually thought that KSHV stole this receptor from the human genome, as it is homologous to human chemokine receptors. Next slide, please. So not much is actually known about what this receptor does inside the cell. So humans have GPCRs as well, and we know that our GPCRs must move around within the cell and interact with all sorts of proteins to signal and fully perform its functions. And I will investigate how KSHV's viral GPCR moves around within the cell, how that affects its signaling, and also what human proteins it interacts with that are important for this function. Next slide, please. Now, I do want to highlight that all of this did take a village. And although you see a lot of names listed here that I want to acknowledge, I do want to give special attention to the College of Science and Engineering. Thank you for awarding me with this fellowship. And I do want to give a very special thanks to the ARCS Foundation because your donation and the money that you have given our university is what's keeping me and my father afloat during the pandemic. Now, the pandemic affects all of us differently, and the ARCS scholarship is really the main reason why we're able to pay bills on time during these challenging times. So thank you so much.
Thank you so much, Jan. That was wonderful. And thank you for representing some of your early work and how much you've accomplished in these last few months, especially in a remote situation. I think it would be really great to maybe talk a bit more about the movement of the GPCR that you spoke about. Why are you interested in that particularly, and how might that alter what's happening in infection?
Yeah, that's a great question. So I talked a lot about how GPCRs have to move around within the cell and why we're studying this in the viral GPCR. So what I didn't expand on fully was how GPCRs were once thought to, do their thing and signal at the surface of our cells, but it was recently discovered that they actually internalize and perform highly specific functions within the cell, and that moving around inside the cell is actually important for their signaling and for regulating our gene transcription. And we're not really sure if the viral GPCR does that and how it does that, or what proteins are important for doing that. So that's going to be the focus of my research for the next 2 years.
Wonderful. Thank you so much, Jan. I'm excited to join you on the journey to figure these questions out. It is my honor to introduce our next speaker, our next faculty member, Dr. Charlie Sakari, who will be introducing our next fellow. Charlie.
Thank you, Erica. So I am honored to introduce our next speaker, Jessica Agnos. So Jessica came to our department via a rather unique path. Her undergraduate degree is in communications with an emphasis in TV and film production. After she moved to the Bay Area, she began taking astronomy classes at City College of San Francisco, where she realized her love for astronomy. She then began working at the California Academy of Sciences as a presenter in the Morrison Planetarium, where she was able to combine her love of astronomy with her passion for public communication. In 2018, she joined our MS program here at San Francisco State and has quickly emerged as a leader in our department. She serves as an officer in the Women in Physics and Astronomy Club. She has organized a virtual tutoring program for a local elementary school during COVID, and she is a co-founder of Astronomers for Planet Earth, a grassroots international organization to advocate for solutions for climate change. We are extremely lucky to have Jessica in our department, and I'm also very grateful to have her in my research group. She will now tell you about her MS research on the chemical compositions of stars and how that information can be used to probe the assembly history of the Milky Way galaxy.
Thank you for the introduction, Dr. Sakari. Being in your research group has been a great experience of growth, and not just as a student, but as a person. And as she said, I'm Jessica Agnos, master's candidate in astrophysics. My research for my thesis is in galactic archaeology, where I study the chemical composition of stars to get clues about their origins so that we may better understand how our galaxy was formed. Now, sometimes when I tell people I'm majoring in astrophysics, I get some strange looks or comments. When I say my research is galactic archaeology, I don't even get a response beyond a wide-eyed stare. So, and if you're like my husband, you imagine me dressed as Indiana Jones running away from a planet rolling towards me, and that's a true story. Now, the phrase, "you are star stuff," was made famous by Carl Sagan. Another phrase of his that resonates with me is, "to make an apple pie from scratch, you must first create the universe." Now, both of these make the same point. Stop and look around. Think of everything you see. Think of you, what you're made of. You are a collection of atoms and elements created in outer space that came together in a massive ball of gas and dust that exploded under the weight of its own gravity to form our sun, and some of the leftover stuff clumped together to form the planets and eventually you. So we are sun scraps. Now, archaeology is the study of old artifacts to piece together history, and that's exactly what we're doing here. Ancient stars give us insight into the happenings of the early universe. Next slide, please. Now, do you know why gold and platinum are so rare? It's because the amount of energy it takes to create those elements isn't produced through the normal fusion that happens inside of stars. Something truly cataclysmic must happen. And also, what are we even looking at? Well, it's starlight. Every single element has its own code embedded in the light that we receive from stars, so we can tell what the stars are made of. So let that roll around in your brain. The elements that we have here on Earth are made in various high energy events in space, and we have the ability to discern what stars have what elements by looking at their light. And all of that information helps us figure out how the galaxy was formed. So plain and simply, I'm fascinated by this, and I feel privileged that I get to spend my time on this planet studying such things. Next slide, please. Now, the perspective astronomy lens is also crucial to understanding how special our planet is and how hard we must work together to preserve it. So this perspective led me to co-found Astronomers for Planet Earth with Dr. Sakari, along with several others from SF State and a few from Yale. We're now an international collective of astronomers working to combat the climate crisis. The day I complete my master's will be bittersweet. I'll be so happy to continue my journey, hopefully onto a PhD, but I'll leave a piece of myself here in this department that nurtured me and embraced my family to come along on this adventure. I'm so grateful to have faculty and administration that value people of diverse and non-traditional backgrounds. And most of all, I'm grateful that my 3 children are witness to it. So thank you, ARCS donors, especially Miss Arlene Inch, for contributing to this journey of mine. You've impacted me and my family immeasurably.
Wonderful, Jessica. Thank you so much. So a question for you. What kinds of events create these heavy elements?
So mostly the observational evidence supports neutron star mergers, and that is what I'm particularly interested in. But some also think that the core collapse supernovae can also create heavy elements. However, more observational evidence is needed for that.
Awesome. I guess we'll know the answer after your MS thesis. All right, so next we will hear from Lily Chen in biology.
Good evening, everybody. I'm Lily Chen, professor in the biology department and chair of Jessica Bryan's master thesis committee. Jessica, a brilliant young scientist, as you will find out, developed a strong interest in cancer immunotherapy. She is very excited to share a preview of her innovative cutting-edge research conducted at the Cell Design Institute at UCSF. Welcome, Jessica, and take it away and share your story.
Thank you for the kind introduction, Dr. Chen. Good evening, everyone. My name is Jessica Briones, and I'd like to start by sharing a little bit about myself. Next slide, please. I was born and raised in San Francisco, and I'm currently a second-year master's student in the biology department. As the first in my family to pursue science and a graduate degree, I appreciate the opportunities and support that San Francisco State has provided in my journey as a scientist. I am conducting my research in the lab of Dr. Wendell Lim at UCSF, and my work aims to improve the standard of care for cancer. In my next slide, you'll see how current treatments like chemo and radiation often impair a patient's immune system. My research hopes to use a different approach by building a smarter, stronger immune system to fight tumors. Cancer is often undetected by the body. If immune cells don't see the cancer, it leads to metastases. So by engineering T cells to express receptors that bind to markers on cancer cells, a patient's immune system can be trained to recognize cancer. I like to think of it as giving T cells a pair of glasses to better see the target. In my next slide, you'll see that by giving T cells this pair of glasses to see and sense the tumor, it can respond by killing the identified abnormal cells. I hope that by testing this therapy in mice, my lab and I can better understand how this may be translated to clinical trials. It is a visceral feeling to reflect on how my initial fascination with cell biology led me to this project with the potential to treat glioblastoma. Research helps me impact the lives of others in a meaningful way, and I am encouraged to continue being a scientific explorer. I deeply appreciate the support that ARCS has given me during my master's. The scholarship has not only provided me with a sense of security during these uncertain times, but it has given me the financial independence to really focus on my research. Having funded my undergraduate degree with student loans and part-time jobs, I am extremely thankful that your kind donation allows me to spend my time doing what I am passionate about, biomedical research. I would like to pursue a PhD after my master's, and I am happy to share that I submitted my applications for programs last week. And because of the ARC scholarship, I was able to apply to every program on my list without worrying about how to cover the application costs. I am excited for my next step, and as an ARC scholar, I want to say from the bottom of my heart, thank you for supporting my dream.
Excellent talk, Jessica. So how do you think your research working from the bench in the hood is going to translate to the clinic?
Yeah, I'm really interested in studying the therapeutic duration of these T cells. I believe that by testing how long these T cells can fight the tumor before they become exhausted, it will inform future studies of the frequency of T cell infusions required to overcome cancer at different stages.
Okay, excellent. Okay, now I would like to introduce my colleague, Professor Kimberly Seashore from Mathematics Department.
Thank you, Lily, and thank you to the ARCS members and donors for your generosity and your support. I'm an assistant professor in the math education in the math department at San Francisco State. Let me just make sure, there we go. And I was an ARCS scholar in the math department in 2007. And because of that scholarship, I was able to engage in research in pure mathematics. That experience was pivotal in my career and enabled me to return to San Francisco State as a faculty member after earning my PhD in math education at UC Berkeley. I'm delighted to introduce Luis Perez, a first-year student in the Math Master's program who comes to us from California Lutheran in Thousand Oaks, California. Luis is pursuing his interest in coding theory, an area of mathematics that is essential in error correction and in the transmission of digital information. In addition to being an ARCS scholar, Luis is a fellow with both the RISE and BAM programs, two programs designed to mentor outstanding scholars from underrepresented backgrounds into PhD programs with the goal of diversifying the STEM professorate. I'm delighted that through BAM, I have the opportunity to work with Luis while he pursues his research interest with his research mentor, Matthias Beck. I'll turn it over to Luis to tell you more about his work.
Thank you for the introduction. Good evening, everybody. My name is Luis Perez. And the next slide, I present to you a little bit about me, which Dr. Seashore mentioned. I am a first-year graduate student pursuing an MA in mathematics. I am a first-generation college student and an underrepresented minority in mathematics. My academic and career goal is to earn a PhD in mathematics and become a professor, where I will have the opportunity to teach, conduct research, and take an active role in diversifying mathematics. So in terms of my research interests, while taking a discrete mathematics course as an undergraduate, I became interested in error correcting codes. Generally, error correcting codes concern the reliability of information. The main problem error correcting codes addresses is guaranteeing that information is received without defects. What I particularly like about error correcting codes are the neat applications they have to other situations, like magic tricks and strategies for games, which can be explained through mathematics. So in the next slide, I provide two illustrations of some of the applications that I've seen before. The one to the left is a magic trick. It's called the parody card trick. The idea is that the girl can correctly guess the card that the boy flipped from the grid using elementary error correcting code concepts. And this is the illustration on the left. Another application called the hat problem is represented on the right. The hat problem is classically set up with three prisoners who are wearing hats that can be of two colors, in this case, white or black. The conditions are that a prisoner does not know the color of their hat, but does know the color of the other 2 prisoners' hats. The 3 prisoners are given their freedom if at least 1 prisoner can correctly guess the color of their hat, and no prisoner guesses the color of their hat incorrectly. And prisoners are allowed to pass if they choose. It turns out that by applying a strategy which comes from topics and error correcting codes, the chance of success jumps from 50% to 75%. The strategy which gives 50% is having 2 prisoners pass and just having 1 prisoner attempt to guess the color of their hat. As an incoming graduate student, I'm in the process of pairing with a research advisor and hope to start working on a project in the summer. I would like to get into the mathematical areas where there are connections with error correcting codes. A specific area I am learning about is combinatorics. I hope to study problems in combinatorics and connect them with topics in error correcting codes. I would like to thank the ARCS donors. Through your efforts, I am in a position where I can focus on research and develop to become a competitive applicant for a PhD program. I can spend more time on classes and research as opposed to looking for ways to fund my education. Thank you.
Thank you, Luis. That was great. I know that you have an article in the final review stages in the Rose-Hulman Undergraduate Mathematics Journal. Can you give us a sneak preview on that article?
Yeah, so this was an extension of the parody card trick that is illustrated here on the left. I was during that time I spent doing research, I was thinking of an application that I can maybe use that has come up, like the parody card trick. So I created a new version of it, which I actually hope to present when we break into breakout rooms. So hopefully Dr. Seashore and I can perform this magic trick.
That's wonderful. I look forward to it. I turn next to my colleague, Jaime Chavez in the Environmental and Conservation Biology to introduce our next ARC scholar.
Hello, good evening, everyone. I wanted to share with you first that I'm a proud former master's student at the Department of Biology at San Francisco State, and now as an assistant professor in the same department. It's an honor for me to continue with the tradition of serving a diverse group of early scientists entrusting their careers. And one of them is Pune Kalhuri. Pune obtained her undergraduate at UCLA, where she double majored in microbiology, molecular genetics, as well as in ecology and evolution. She has already co-authored 2 scientific publications where she merged these fields, in particular in the area of conservation genetics, applied to species of conservation concern in North America. Pune will expand on these research fields in my lab as a master's student, where she will expose and explore these research to species of birds found on the Galapagos Islands here in my background. So Pune, please take it away.
Thank you for the introduction, Jaime. Hi, everyone, and thank you for joining us this evening. I'm going to be talking a little bit about my project on a species response to future climate change on the Galapagos. So I've known that I want to study ecology for quite some time now, ever since I took my first environmental science class in high school, really. And while I was an undergrad at UCLA, I had the opportunity to do field research in French Polynesia, where I witnessed some amazing biodiversity on the coral reef ecosystems, but also the devastating effects of human impacts on one of the most biodiverse ecosystems on the planet. I later worked on a conservation genetics project studying sea otters, which were a species that were nearly hunted to extinction by fur traders in the 17th to 20th century. I decided that I wanted to continue doing research in conservation genetics. This time I decided to choose a new species of bird, the Galapagos yellow warbler. So some of you might be familiar with this bird, especially if you've been bird watching. The yellow warbler can be found across North America, but the Galapagos yellow warbler is a subspecies that is endemic to the Galapagos. and it can be found on all of the islands in the Galapagos, as well as across elevations from sea level near the coast all the way up to 5,600 feet, which is the maximum elevation on Wolf Volcano on Isabella Island. With climate change, a lot of landscapes are getting warmer and species are finding themselves in new habitats that might not be suitable for them. So this leaves them with really three options: either adapt, find a new habitat, or die. And a lot of species respond to global warming by shifting their distributions towards cooler climates. On an island landscape, there isn't really anywhere to go other than up. And because these yellow warbler populations are found across elevational gradients, it's possible that these birds show local adaptations depending on what elevation they're found at. So we're left with the question, how will birds at different elevations respond to climate change? Will some populations be more likely to persist compared to others? And my project uses DNA samples to find genes that are related to elevation in these birds, and then uses this data to predict the potential for yellow warblers to persist in warming climates. And hopefully this research can be used to inform conservation and management of the species in the future. I wanted to give a huge thank you to the ARCS Foundation and the Betty and Bruce Alberts and Ray Benton Family Fund. Their scholarship is allowing me to pursue a graduate education in a field that I've always hoped to study, but I worried wouldn't necessarily be possible. And I feel incredibly grateful to be pursuing an area that I'm so passionate about. Thank you.
Thank you, Panay. That was great. I have questions for all of us who love birds in North America. What makes this bird in the Galapagos being so unique now that we're talking about being the same species?
So that's a great question. And as you mentioned, this bird does look a lot like the bird we find on the mainland in North, South, and Central America. But the bird has been found to be genetically distinct, even though it's fairly recently colonized the Galapagos. And this is really important. In conservation biology, there is a concept known as an evolutionarily significant unit, which is considered a group that is considered unique enough to receive special conservation attention.
Thank you so much. I would like to introduce to Ellen Hines from the Department of Geography and Environment.
Greetings, everybody, and thank you so much, ARCS donors. This is Rebecca is my second student who has got an ARCS fellowship, and I've really seen how much difference this makes in my students' lives. I'm very pleased to introduce Rebecca Lane. Rebecca has joined my lab from Kansas, where she completed her BS in biology and a Master's of Science in Education, and somehow knew she wanted to come and study the ocean. Before coming back to school, Becca taught 9th through 12th grade field ecology and science for English speakers of other languages. With her project, Becca has initiated wonderful collaborations with the Marine Mammal Center in Sausalito, NOAA, and the Anthropocene Institute for this project that integrates cutting-edge ship tracking technology. Her research has created methods and will create results that will contribute directly to management planning in the Greater Farallones National Marine Sanctuary to minimize shipping collisions with baleen whales. Please take it away, Becca.
Thank you so much, Ellen. It's such an honor to be in your lab. Good evening, everybody. I'm excited to be here today with you all for the ARCS Awards celebration. Today, I will briefly discuss my research, my passion, and my gratitude for this scholarship. The title for my research is Whales in a Highly Urbanized Estuary, Evaluating Risk of Ship Strike to Humpback Whales in San Francisco Bay. Humpback whales have always fed off the coast of Central California. In fact, here we have two distinct population segments that are listed under the Endangered Species Act. However, in 2016, local researchers started observing these same humpback whales feeding inshore, east of Point Bonita and even east of the Golden Gate Bridge. And they've been back every year since. It's great to be able to see humpback whales from the Golden Gate Bridge or the headlands, But this new habitat shift does put them in close association with human activities, particularly shipping. Ship strike is a major cause of death for large whales, especially in places where whale habitat and human activities overlap in space and time. Advance, please. My research project aims to create a risk assessment for ship strike to prioritize management for humpback whales in the bay. To do this, we were using whale sightings data from 2016 to 2020 from the Marine Mammal Center. We're filtering the data to include the precautionary zone, which is shown here in orange, as well as San Francisco Bay and the area excluded from the sanctuary system. I will also acquire vessel data from two sources. The first is the Automatic Identification Systems, or AIS, which is required on most commercial vessels. This is open source data that can be found online. Second, I will use a marine monitor, which uses a marine radar and a high definition camera in order to track vessels in the bay that do not use AIS, like recreational vessels. Next slide, please. Using this data, we can create a more complete habitat risk assessment model, kind of like the one shown here from Redfern et al. 2020. However, our model will include whale sightings inside of the bay since 2016, and will be a more comprehensive view of vessel traffic since it will incorporate recreational vessels as well. Next slide, please. I've been interested in the great whales and how we can protect them since I was a little kid living in this gray house. At the time, it seemed like a really lofty dream, and here's why. I wasn't anywhere near the ocean. I also come from a single parent household, which made finances tight. It was an extra challenge to fund myself during school so that I could travel and so that I could get the experience necessary to advance in marine mammalogy. But I continued to pursue my goals, applying to internships around the US, gaining research experience, and eventually teaching high school biology full-time so that I could spend my summers pursuing unpaid marine mammal internships and save up money to move to the coast. Next slide, please. Now I'm here at San Francisco State, and I can't begin to express how thankful I am for the support you've given me so that I can continue to study and protect these magnificent animals. My goal is to become a PhD marine mammalogist, studying anthropogenic impacts to large whales like entanglement and ship strike, while also mentoring my own students. And the last slide. Thank you. Your generosity is supporting my passion and my research, and it's helping me achieve my childhood dreams. I cannot thank you enough.
Thank you so much, Rebecca. How many baleen whales die of ship strike every year, and how does this affect their population sustainability?
That's a great question, Ellen, and especially important since we're talking about the fact that these humpback whales that come here are endangered species, actually. So between blue whales, fin whales, and humpback whales on the West Coast, we have about 80 whales that die each year. And particularly for humpback whales, that's about 2 times the limit for biological removal so that the population can be sustainable. So the answer is too many whales.
That's for sure. Well, excellent presentation. Thank you. I'd like to introduce Professor Edward, excuse me, Professor Edward Connor from the Department of Biology next.
Thank you, Ellen. It gives me great pleasure to be able to introduce my graduate student, Stephanie Singh. Stephanie Singh plays a leadership role in my lab because she organizes her lab mates to accomplish numerous research tasks that we need to accomplish, but she also helps foster a supportive and interactive atmosphere. Stephanie's research focuses on how plant-eating insects may be manipulating plants to make them more nutritious for the insect by inhibiting the defensive responses of the plant, and in some cases, inducing a tumor-like growth called a gall to house and protect the insect. Take it away, Stephanie.
Thank you, Ed. Good morning, or sorry, good evening, everyone. My name is Stephanie Singh, and I'm currently a second-year master's student in Dr. Edward Connor's lab. A couple of things about me. I'm a first-generation Asian American and the first to pursue a science degree in my family. I graduated from SF State with my BS in cell and molecular biology, but felt very supported within the college, so I decided to also pursue my master's here. While I was an undergraduate doing my studies in cell and molecular biology, I was introduced to ecology and evolutionary biology, and I quickly fell in love. As a result, I joined Dr. Edward Connor's lab, in which we study plant-insect interactions that lead to the formation of plant tumors called plant galls. Within this fostering lab environment, I am able to intersect my cell molecular biology background with ecology and evolution research topics. Next slide, please. Our research aims to answer questions related to how insects can cause these abnormal growths of plant tissue. The picture on the left shows a picture of our plant gall, in which our fly, shown on the right, feeds in the larval stage and further develops into the adult stage before emerging. I am involved in several projects that explore the hypothesis that these insects are creating plant growth hormones in order to manipulate their host plant into forming these galls. I am examining various biosynthetic pathways involved with these plant growth hormones and determining the localization of these hormones within the insect. We are using a mix of fieldwork, bioinformatics, and wet lab work in our projects. We believe that there is widespread manipulation of host plants among plant-feeding insects by use of a mixture of different plant growth hormones. Next slide, please. I'm very fortunate to have the opportunity to apply for PhD programs. Just a few years ago, I didn't even know what that meant, but I've had just submitted for various programs around the country. My eventual goal is to become an academic scientist at a higher education institution, where I can continue to pursue my passions for teaching and mentoring students while also continuing research. Because I've had an amazing amount of support, mentorship, and personal growth in my journey, I want to give back to the scientific community by providing the same to future generations. Next slide, please. I would like to thank my advisor, Dr. Edward Connor, and my lab mates, pictured here in our annual Halloween lab picture. I would also like to show my deep appreciation for my funding sources that have supported me through my schooling and research. Before the COVID shutdown, I was working 2 jobs to help support my younger brother and myself. However, that has changed since the shutdown, and ARCS has really made a difference to help me through these tough times. So to our donors, thank you for supporting students like myself to give us a better chance for our education.
Thank you, Stephanie. Very interesting. I know you touched a little bit about some of the techniques you used in the research you're doing, but could you briefly describe sort of the different components of your research work? I want everyone to kind of understand the variety of methods and techniques that you've had to integrate in order to accomplish your project.
Yeah, thanks for the question, Ed. So within my research, I use a variety of techniques and methods in order to explore our lab topics and answer our research questions. Some of this includes field work, such as flying out to Minnesota to an arboretum to collect our plant specimens. We do some greenhouse work where we grow our host plant, and we also rear flies to induce these plant tumors within our greenhouse. I do a fair amount of wet lab work, including RNA extraction, reverse complementation, qPCR, as well as antibody staining. We also use microscopy techniques, including microdissection of flies and confocal microscopy. And lastly, we also do computational bioinformatic work, including DNA sequence analysis and primer design.
Thank you. And now I'd like to turn the proceedings back over to our Dean, Carmen Domingo.
Thank you so much, Ed. Wow. Congratulations to all of the ARCS honorees. It's, you know, like if we could unmute and like, woohoo! It's so impressive. You know, you all are doing just a fabulous job. And we're just truly impressed with what you've accomplished and have no doubt that you're going to push the scientific field forward in really exciting ways. So now it is my pleasure to ask Leslie Van Houten, Co-Vice President of University Relations for Northern California Chapter of the ARCS, to share a few words with us.
Oh, thank you so much, Carmen. Wow, that's all I can say. As you know, this is our first presentation of our virtual scholar celebration, and it was absolutely spectacular. Dean Domingo, you assembled an incredible team of faculty and scholars, and you bring such energy, dedication, and enthusiasm to your work and to the mission of San Francisco State. We at ARCS are thrilled to be able to work with you and your remarkable team. And I have to add my shout out to Lonnie. We wouldn't have been here. We wouldn't have had this fabulous program without her. Clearly her talent and hard work paid off. And our faculty members, the faculty mentors, we know that you are playing it forward. You all are a part of a long line of scholars who have explored our world, added to the scientific knowledge, and then mentored your students, passing along your love of exploration, creation, and lifelong learning. Thank you so much for the role you played here. And then not least of all, our scholars. I have to say, some of you brought tears to my eyes. Of course, it's not hard, but you're just amazing. You are our future scientists of our nation, and we know we're in good hands, and that is really rewarding and really comforting. Thank you for sharing with us your journey into your piece of the scientific world, and know that we at ARCS are amazed and thrilled to be some small part of your journey. Kudos to you, and I know that like your mentors, you will play it forward when you have the opportunity. Thank you. And now I want to pass this back to Joe, who's going to give a couple of important announcements.
Well, gosh, I really do second all of those kudos, both to the scholars, the PIs, and the leadership of this College of Science and Engineering. And it does bring tears to our eyes, and it's not that easy with me, but honestly, you make us really, really proud. I just wanted to take one more moment to invite you to join us for a Zoom webinar this coming Thursday from 6 to 7 p.m. It's put on by our Science Activities Council, and it features a professor of otolaryngology, that's head and neck, at UCSF. His name is Dr. Daniel Knott. He actually is a wonderful researcher and surgeon who pioneered a technique of cross-linking collagen with hyaluronic acid. And that technique has enabled him to do transfers of facial nerve muscles to correct deformities that are related to congenital deformity and also traumatic injuries. Ann Thorson, who's a cardiologist and a member of ARCS, is going to introduce him. And I promise that it will teach us all something about the importance of basic research, which then becomes the foundation of meaningful translational advances. And I am inspired by so many of you wanting to continue to teach. And I'm also inspired that so many of you are already engaged in teaching, particularly with students who are underrepresented in STEM. It must be the contagion of Dean Domingo. That's all I can say. So thank you all again. We just loved hearing from each and every one of you.
Thank you, Joe.
And I'm going to give it to Lanny, right? Or Carmen?
I will tell everyone what's next, because the formal part of our event is over, but we thought it might be nice to be able to just have a little informal part. You know, normally we would be gathering with our drinks and having a conversation around the table at one of your lovely hotel events. So what we're going to have is we've set up 9 breakout rooms. It would have been 10, but as you heard from Jessica, she has to go to an event this evening. And it's very easy. We're going to set up the breakout rooms, and you don't have to do anything. You don't have to touch your computer or anything. You are going to be randomly sent to one of these 9 breakout rooms. And in that breakout room will be an ARCS scholar along with their mentor. And in some cases, we encourage the ARCS scholars to invite their families. So you might have the delight of meeting some of their close and biggest supporters. And then when it's over within the room, then you won't be coming back to the main room. This way, we know it's close to dinner, and so in any case, it'll, you know, as much time as you want in the breakout rooms, it'll be moderated by the faculty member there. So I just want to thank everyone for really a lovely event, and the ARCS scholars for their wonderful presentations and all the wonderful words of encouragement that everyone shared. So I think I'll turn it over to Joe. I don't know if that was a crashing computer, but Joe, why don't you go ahead and get everyone sent to those breakout rooms. And I wish everyone a wonderful holiday if we don't see you before that, and hopefully maybe at the ARCS event this coming Thursday.